All right, welcome everyone. Welcome, welcome, welcome. You are beyond welcome. So glad to see you here today. Looking forward to this class. It is going to be a very good, exciting, wonderful class. This is going to be on the mitochondria again. We're going to be talking about the mitochondria, what is going on inside the mitochondria itself, dealing with specifically with the Krebs cycle, which is such an important point. And so this is part 3 of the mitochondria. Part 1, many of you already saw, which was dealing with creation, evolution, and how we got to the mitochondria, which is so important for you to be able to respect the mitochondria. Part 2 was the mitochondria, and then dealing with outside of the mitochondria, which is dealing with glycolysis, which is super important to be able to understand the Krebs cycle. So we're here today on the Krebs cycle. It's going to be extremely powerful course. Before we get started, though, I'm going to just lay down some ground rules, and then we're going to get into the disclaimer, and we will move right into it. So first of all, for those of you that are here, please make sure that you have subscribed. If you can, put a 1 in the chat so that I know that you can hear me. If not, if you can't chat, please subscribe. It allows you to be able to chat. It also will remind you when we have a class as well. Every time we go live, you will get notified if you decide to do that on your settings in your YouTube. Okay. And then also the other thing is replays of these, the full replays will be available on our Patreon, which is a subscriber base, but you can watch it now for free. So make sure you take advantage of watching this and getting the information that you can. But I always advise people to rewatch videos so that you can get the full knowledge, understanding, and information. With that, we're going to move forward into our disclaimer, and we'll be right back.
Disclaimer. The information contained in this video is intended as an information resource only and is not to be used or relied upon for any diagnostic or treatment purposes. The video's information does not create any patient-physician relationship. Dawood Yisrael makes no representations about the accuracy or suitability of this content, as scientific and medical information updates and changes daily as more substantial information is discovered. Dawood Yisrael and any business associated during this video shall not in any event be liable for any direct or indirect damages, losses, injuries, or liability whatsoever suffered as a result of your use or misuse of this video's content. Use of this video is at the viewer's sole risk.
Yeah, Go ahead.
Being here is he's been bigger than I could even articulate, and I think this brother's chosen to share a gift that he's been blessed with. I already knew I was the most ignorant person, but this brother never judged me. I came clean with my errant ways, never felt judged, and I'm leaving feeling better on so many levels, and I'm so thankful for him and his peers that showed up. So I'm looking forward to being back next week to gain some more knowledge, and I can pass that on to my grandchildren and hopefully anyone in my circle. But I'm going to grow with him if he allows me to, and I'm grateful.
Absolutely. You grow with it. All right, so I have another beautiful blessing. My books, my physical book is back in. I've got it in. You can always order it on Amazon, which is in the link in the description of this video. However, I've got it, the book itself, which I'm so happy about. I got them in. So if you're in the Chicagoland area, please make sure that you hit me up on my Patreon, or you can put it in a text here that you would like to get my book, and or you can contact me at 708-549-4892, and that will allow you to be able to get a copy of my book. I will sign it for you as well, so you can get an autographed copy of the book. But it's an excellent book. It has the entire book with questions and answers in the back, which is extremely powerful because it helps you to retain the information. Also, if you're going to take my health class, which the link is also in the video description below, if you're going to take my health class, then I advise you to definitely have one of my books. But when you take the health class, you can also get the electronic version of my book for free. It comes with the class, but most people want the physical copy. So I just advise you to get it early. And then if you have any questions or anything at the class, you can do that as well. I'm considering just making this next class an online class because I wanted people from all over the country to be able, and around the world, to be able to be on that class. But that's a decision I haven't made totally yet. But so either it'll be physical and online, or it'll be online only. Okay. So with that, do we have any questions before we begin? If not, I'm about to jump right in and get started. I know yesterday we had a couple people on the question and answer call stating that the first two went over their head. So I apologize for that if it did go over your head, but there's many people that are watching that'll really be able to retain and get the information from it. So I'm going to continue today, and then I'll do a simple breakdown on Wednesday. I'm going to do a simple breakdown of everything that we talked about on Wednesday, but I had to get this out. It's a lot of good information. As a matter of fact, there's so many things inside of this will help a person be able to reduce or stop or allow their body to heal cancer as well, which I'm seeing as I go through this process. So let's get started. Let's make it happen. Let's see what the mitochondria Krebs cycle is all about. Okay. So with that, the Krebs cycle itself, let's look at it. Now, let me see if I can make this bigger for you all. Yes, I can. All right. So it's a little bit bigger. The Krebs cycle, also known as the citric cycle, is a series of biochemical reactions that occur in the mitochondria of almost all living cells. So almost all living cells do have this process, okay, the Krebs cycle. The cycle's primary purpose is to produce high energy molecules such as ATP, NADH, and FADH. This is the energy. That's why they call the mitochondria the power of the cell, right? It's the power plant of the cell. And so the Krebs cycle is like multiplying the power that we talked about with glycolysis. Okay, it takes what glycolysis does and multiplies it. Okay, and which is extremely powerful, which is so important for your body to get the proper energy that it is supposed to have. Okay, if your body doesn't walk through this Krebs cycle, there's a major issue, right? There's a major issue going on in the body. Okay, and we're going to discuss that today. So, the Krebs cycle. Why is the Krebs cycle essential for life as we know it? All right. The Krebs cycle is a sequence of 8 reactions. There are 8 reactions that occur in most living cells to produce energy. Carbon dioxide is released, 1 ATP molecule is created. NADH plus is reduced to NADH 3 molecules, and 1 FADH2 is formed from FAD. We're going to get deep into that, so don't worry. It's okay. I just want you to get that what's happening in the Krebs cycle is that there's 8 chain reactions that are going to produce ATP, NADH plus and FADH2. Okay. All right, or NADH rather, and FADH2. That is very important to get, okay? So that's what I want you to majorly understand, because this is your energy. You think it's just ATP, but it's not. It's ATP, NADH, and FADH2, okay? And I want to thank, I want to definitely give thanks, thank Jerry for bringing this subject up. There was so much I learned just from studying this subject and getting this class prepared for you all. So I greatly appreciate that for being a clearing and bringing that so that I could get this information, because it's extremely powerful. So you are right this minute the site of some unbelievably complicated biochemistry, okay? So you are a what? Unbelievably complicated biochemistry. You yourself, you're an amazing being. You are beautifully and wonderfully made. Literally. That's what they're saying. In order for your body to do literally anything, jump on a trampoline, walk yourself to the bathroom, move your eyeballs as you read this article, you need to be able to accomplish something called cellular respiration. And if you remember, if you've been following me, you know that in the Zenzino video, which is up, right? You can watch it. It's very important. I talk about cellular respiration and the omega-3s and why it's so important. And I'm really, the more I study this mitochondria glycolysis as well as the Krebs cycle, I understand exactly why the balance oil works the way that it does. I understand it totally. And so I want you to understand that it is extremely important if you have the balance oil that you take it. It's extremely important. It's not a light thing. It's super important, okay? So I want you to get that you need to be taking the balance oil on a regular basis if you have it. If you don't have it, please make sure you contact me. I think I have the link there in the Zenzino video for the for the balance oil. It's extremely important. And the Xeno gene once every quarter in the beginning until you get totally cleansed is important to get rid of all the senescent cells. And you're going to understand why when we get done with this video. So you need to be able to accomplish something called cellular respiration, in which your cells create energy out of oxygen, right? You breathe and the food you eat, okay? It's important. This is extremely important, right? Because oxygen is what? Spirit. So what they're saying is you have to be able to get the spirit out of the food that you're eating, okay? You need to be able to accomplish something called cellular respiration, in which your cells create energy out of the spirit or oxygen or ruach or air you breathe and the food you eat. This is why when we talk, I'm always telling you eating is spiritual. Why? Because you have to be able to lower the amount that you're eating to allow more spirit in the body, which is going to cause cellular respiration, which is vital to your life. You can't do nothing without this, as is stated earlier. And as you can imagine, turning a peanut butter jelly sandwich into a push-up is a bit of a process. Now, I'm telling you, do not eat peanut butter and jelly sandwiches. Please do not do that. Right? But just get the understanding that peanut butter and jelly sandwich, because our body is so amazing, it can take negative things we put in the body and turn it into power. Right? But you're gonna hurt something in doing so. Okay? But it is a process. So let's see how the Krebs cycle makes this scientific happen. Now, cellular respiration. One main goal of cellular respiration is to create a specific type of stored energy called ATP. This is very important, okay? One main goal of cellular respiration is to create a specific type of stored energy called ATP. And this is what your body's doing all day long. Or adenosine triphosphate. Remember, it's 3 phosphates. ADP is what? 4 phosphates. 2 phosphates. It's right? Diphosphate. So it's 2 phosphates, not 3. Which means ATP is stronger. Okay? Think of it as the energy language spoken by your cells. Sunlight is energy, but we can't power our bodies with it because it's not speaking the energy language our bodies knows. However, if you study Elitam Elamine book called Soul Transformation, human photosynthesis. I believe it's called human photosynthesis. He's teaching you how to be able to allow your body to turn sunlight into energy. See, your body can transform into different forms and take different forms of food when necessary, or when you learn to utilize it properly. So we can take sunlight into energy, but you have to learn how to do it. And this is the breatharian journey. So even though they say that it's possible, is what I'm saying. Because it's not speaking the energy language our bodies know. Animals' bodies only speak ATP. And that's true for the majority, because we don't know how to do the soul transformation or understand human photosynthesis. Okay? So one step of the long road from sandwich to push-up is the Krebs cycle, also known as the citric acid cycle or the tricarboxylic acid cycle, the TAC. That's what they call it. A lot of times you'll see it as TAC. They're either going to say citric acid cycle or it's going to say TAC. Okay, named after Hans Krebs. He was the first to work out this piece of biochemistry in 1937, and as a result, he won the Nobel Prize in Physiology of Medicine in 1953. All right, it was well earned because the Krebs cycle was extremely complicated, using changes in chemical bonds to rearrange energy. So it's rearranging energy. The Krebs cycle happens in our cells across the inner membrane of the mitochondria. Now, if it was that, this is why I made the video on creation evolution, right? And dealing with the atomic structure of the universe, dealing with the mitochondria. Because if you think that it was amazing for him to find it, just think of how amazing it was for that system to be created. It took billions of years to create the mitochondria and the Krebs cycle and all the things that we're gonna be talking about today. I want you to understand how powerful this is, right? It's not a light thing. It was well earned because the Krebs cycle is extremely complicated, using changes in chemical bonds to rearrange energy. The Krebs cycle happens in our cells across the inner membrane of the mitochondria. So in other words, we're finally into the mitochondria, right? I showed you how the mitochondria was developed. It comes all the way from what? Hydrogen all the way up, right? And it's found in the eukaryotic beings, right? Right? So, and then, or eukaryotic beings, excuse me. And so the mitochondrial is where we find it. The organelles responsible for cellular power production. So our power in our cells, which is us, we're made of trillions of cells, that is done through the Krebs cycle. Cellular respiration is a multi-step process, starting with glycolysis, which we went over on last Thursday, I believe it was. Yeah. Or was it? Yeah, last Thursday. Which breaks down the 6-carbon compound of glucose and serves up these 3 carbon molecules called pyruvic acids, right? Those are remember the sugars that were broken down? And they created 3 carbon molecules, right? That's what we went over. And 2 energy-rich compounds called NADH. From here, the Krebs cycle takes place. So it created ATP, NADH, and these pyruvic acids. Okay. Now, the Krebs cycle is an aerobic process, meaning it needs air. Remember, the glycolysis can happen with or without air, but the Krebs cycle, it needs air. Okay. Meaning it needs what? Oxygen or spirit to work. All right. So along with the process of oxidative phosphorylation, the Krebs cycle gets the business right away, mixing carbon and oxygen in the respiration pathway. First, 2 carbons enter the cycle. The 2 carbons are oxidized and removed from the cycle, says Dale Beach, professor in the Department of Biological and Environmental Sciences at Longwood University. We can think of this first step as completing the oxidation of the glucose sugar. And if we count sugars, 6 entered the respiration pathway at glycolysis, right? That happened. We went over that last week. 6, remember, it was a 6-ring compound. If you don't remember that, go back to that video and watch it. It was a 6-ring compound that made up sugar, and it got split during that process into 3 it says it up here, so I want to make sure I say it right. 3 carbon molecules, right? At the end. Okay? So that's what happened. And a total of 6 must exit, though. These aren't really the same 6 carbons, but it does help to reinforce the conversion of glucose into carbon dioxide through the pathway. So here's the issue. What happens is these 6 still need to be dealt. I mean, the other 3 still need to be dealt with. Okay? This is very important, because if it doesn't happen, you're going to have what? A disease. And there are diseases caused when the Krebs cycle is not kicked in, or if your body has a breakdown in the Krebs cycle. Right? So this is we're going to get deep into that in a minute, because we're going to start understanding where these diseases is coming from. One of the carbons off the 3 carbon molecule bonds with 1 molecule of oxygen and leaves the cell as carbon dioxide. CO2 is carbon dioxide. Okay? This leaves us with a 2 carbon molecule called acetyl-coenzyme. Now, acetyl-coenzyme, this is like a coenzyme. Whenever you see this YL, that's a coenzyme. A coenzyme helps an enzyme be able to catalyze, right? Or in other words, transform something into something else. That's what it means. It's a catalyst. Okay? Now, this leaves us with a 2 carbon molecule called acetyl-coenzyme A, or acetyl-CoA. This is what they always gonna put it like this. So when you see that CO, that's coenzyme, okay? And they call it coenzyme A. Okay, this ain't alpha, this is A. Further chemical reactions reorganize the molecules in a way that oxidizes the carbons to get another NADH and FADH. So it's increasing the energy. And remember, NADH is stronger than ATP, or more energy, and FADH is also stronger. NADH is the strongest. FADH is right behind it, and I'll show you why in a minute. After completing the respiration pathway, the Krebs cycle undergoes a second oxidation process that looks a lot like a traffic roundabout. It's what makes it a cycle. Okay, so after completing the respiration pathway, okay, the Krebs cycle undergoes a second oxidation process that looks like a traffic roundabout. It's what makes it a cycle. The acetyl-CoA enters the cycle, combining with oxaloacetate to form citrate synthase, hence the name Krebs cycle. Okay, so this right here is where that's step 1, really. Okay, so it's going to do that in step 1, and that's where the Krebs cycle is going to kick in. So it goes from glycolysis into this Krebs cycle, hitting this oxaloacetate, right, to form, or acetate rather, to form citrate synthase. To form citrate synthase. And that citrate synthase, remember, whenever you have the ASE, that's an enzyme. That enzyme is now going to rock, right? So the catalyst, which is the acetyl-CoA, hits this and then hits that. Okay? That's basically what's happening. Hence the name Krebs cycle. The citric acid is oxidized over the course of many steps, shedding carbons all the way. Okay, this is key. Shedding carbons all the way, because these carbons have to be dealt with in your body afterwards. Right? So remember, we have an issue here with the after the glycolysis, these other carbons and stuff have to go into the Krebs cycle. If it don't, it's going to cause a problem. And we got carbons being shedded during Krebs cycle, which if they're not dealt with by the body, then we got problems. And remember, you got trillions of cells doing this every second. You got trillions of cells doing this, creating this, going through the Krebs cycle every second. Right? So shedding carbons all the way around the roundabout until oxaloacetic acid is regenerated by oxidation of malate. And you're going to see this clearly because I got a chart and everything for you on this. Don't worry about it. I just want you to start getting these words and stuff in your head. Don't worry about understanding it fully yet, because you're going to understand it when I get done. As carbons drop off the citric acid, they turn into carbon dioxide molecules and are spat out of the cell and eventually exhaled by you. So you have to breathe them out. That's why breathing is so important, right? This is why you have to have a carbon oxygen balance in the air. That's why trees are so important, right? Super important. Now, energy production and coenzyme A. During the second oxidation, a new high energy bond is made with the sulfur of CoA to produce succinate CoA, says Beach. There is enough energy here that we can directly produce an ATP equivalent. Okay, now, this is important. This is step 2, all right? What's going to happen is GTP is going to be created. GTP is like the RNA for DNA. DNA, you have DNA, which is the original, and then you have copies of the DNA called RNA, right? Which are not the original, and they can't do everything DNA can, but they can do a lot of it. GTP is weaker than ATP, but it can be used by the body for a second to do what it needs to do to create more ATP, okay? So there is enough energy here that we can directly produce an ATP equivalent. GTP is actually made, but it has the same amount of energy as ATP. This is just a quirk of the system. So it has the same amount of energy. I apologize. I thought it was weaker. I don't know why I thought I read something that was weaker. Okay, anyway. So it has the same amount of energy. So the removal of the coenzyme A leaves us with the succinate molecule. From the succinate, and with creation, there's a reason why this is GTP and not ATP. They might not have figured it out, but it definitely is a reason for that. The removal of the coenzyme A leaves us with the succinate molecule. From the succinate point in the cycle, a series of steps to rearrange chemical bonding and some oxidation events to restore the original oxaloacetate. In the process, the pathway first produces a low energy FADH, which is power, right? And a final NADH molecule. Now, I know FADH is weaker than NADH for sure. That's a fact. And you'll see that in a minute. Beach says, implications and evolutionary considerations. For each glucose entering respiration, the roundabout can spin twice, once for each pyruvate that enters it. So remember, there's two. When we went over the glycolysis last week, there were two pyruvates that were made, remember? Both of them have to go through this process. So for every glucose molecule, right, just a little, we eat massive. When you eat some, you eat massive glucose molecules. But one glucose molecule, for each one glucose molecule, it has to go through the Krebs cycle twice. Just like it did on the second half of glycolysis, right? So it's going to go through the Krebs cycle twice. Now, however, it doesn't necessarily have to go around twice, since the cell can siphon off carbons from other macromolecules or put more into the cycle by sacrificing amino acids or capitalizing on the energy of stored fat. That's ketosis. That's ketosis. I'm going to show you something amazing about ketosis in a minute that I learned just from studying this, the Krebs cycle. It's amazing. And I kind of figured it out, so I looked it up and it was true. So I want you to understand what's about that, why ketosis is so powerful. This is why fasting is so powerful. Fasting is the key. When you really understand the mitochondria and how it works, it's the key to everything, right? It's the power of the cell. So every cell is going through this process. Complex biochemistry, but according to Beach, one thing to note about the Krebs cycle is the frequent appearance of adenosine. Okay? Adenosine. It's in NADH, FADH, coenzyme A, and ATP molecules. So it's everywhere in there, okay? Now, adenosine, which I believe is it's amino acid. Adenosine is a molecular handle for proteins to grab onto. We can imagine the evolution of ATP binding pockets being shared and recycled so that these become binding sites for other molecules using the same or similar motifs, okay? Now, let me see. I think that's it on this one, okay? So now we're going to get into the Krebs cycle for real. So I'm going to show you a diagram of it, okay? Let me see if I can make this bigger for you. Oh, yeah, that should do it. There we go. So the Krebs cycle is the cyclic cycle, right? I'm going to go out just a little bit one time. See if we can get that for you better. There we go. So you can see it's a cyclic cycle, right? Now I'm going to go back to the big size. So it starts with glucose coming in or fatty acids. These fatty acids can be muscle, right? Breaking down protein rather, right? Or it can be fat itself, lipids. This is why ketosis is so powerful, because it can skip glycolysis and come straight into the cycle. Okay? Now, the thing that's deep about it is, if a person has cancer, cancer uses glycolysis in order to be able to multiply. If you're fasting and ketones, really these are ketones coming in, ketones, because the fat is going to turn into the fat and the muscle going to turn into ketones. When the ketones come in, because they are going to get they create acetyl coenzyme A, right? Without having to go through glycolysis. This is how other animals that are carnivores, this is how they eat. This is how they don't have glucose. They don't go through glycolysis. Remember? For those of you that have been in a health class, they do it off of fats and proteins. So when you fast and you allow your body to break down the fat and protein, right? Remember I said all disease has to go through this process where it's going to break down, it's going to eat itself, what is called autophagy, and your body's eating it up. This is the process. But it skips glycolysis, which is necessary to proliferate cancer. This is the key. Fasting ain't no joke. Fasting works. And this is why. It is this simple. This is why it works. So when glucose, this is why I keep telling y'all, I will tell everybody, and it doesn't matter. I said everything else, I don't care who it is. I don't care if it's a doctor. I don't care if it's a holistic doctor. I don't care if it's me. Everything else that I tell you to do is just a Band-Aid until you do the long-term supervised water fast. Period. It's a Band-Aid. Because you have to cleanse your whole body again. You have to do a reset. And this is the reset. Right? So acetyl coenzyme A, which we talked about earlier, right, is going to hit what? The oxaloacetate is going to, the citrate synthase and the citrate together. All this is going to create the next phase, which is the isocitrate, right? It's going to hit this, create that, create citrate. Citrate is then going to hit aconitase as well, plus water, or H2O, is going to equal isocitrate, right? So phase one to citrate to isocitrate. Then comes isocitrate is going to become alpha keto glutarate, right? So we see keto is in the midst of this, don't we? Right? And I'm going to break all this down. Don't worry. I'm just showing you the quick route. And then this, the the alpha ketoglutarate is going to have alpha ketoglutarate dehydrogenase, which again is an enzyme, and then it's going to have this. It's going to create succinyl coenzyme A, and then this is going to have its synthetase, right? And then it's going to move into. It's going to create ADP, right? And it's going to go to succinate. And then you see the FADH is created. And NADH was created twice back here, just to show you already. Fumarate, right? Then water's coming in. Then malate. Then NADH, right? And we're back to this cycle again. And then that second coenzyme is going to come through and do it again. Remember, it's 2 for every glucose molecule. So we're going to read through this, so we can really get an understanding. This is really deep. This is deep right here. We already we already know that. We got that. We're going to start here. Krebs cycle is part of cellular respiration. Never forget that word. This is super important. Cellular respiration. Cellular respiration is a catabolic reaction taking place in the cells. It is a biochemical process by which nutrients are broken down to release energy, which gets stored in the form of ATP, and waste products are released. In aerobic respiration, oxygen is required. Don't ever remember that. Spirit is required. Do we have any questions so far before I continue? Because I did go over a lot already, but I'm about to go deep now. So I want you to really, if you got any questions, please feel free to ask them. Again, subscribe if you have not subscribed to our YouTube channel. All right. If you can't ask questions, you will be able to then chat after you subscribe. Okay. And you will also get all the updates of when we're going live. So, cellular respiration is a catabolic reaction taking place in the cells. It is a biochemical process. And you can ask questions during the class too if you want. It is a biochemical process by which nutrients are broken down to release energy, which gets stored in the form of ATP, and waste products are released. In aerobic respiration, oxygen is required. Super important. Spirit is required for that. Glycolysis is not. And cancer loves glycolysis. Though it uses oxygen, it's using up your oxygen. It's starving you of oxygen. And it's doing it on purpose, in my opinion. But, that's how it rolls. Now, cellular respiration is a 4-stage process. In the process, glucose is oxidized to carbon dioxide, and oxygen is reduced to water. The energy released in the process is stored in the form of ATPs. 36 to 38 ATPs are formed from each glucose molecule. Now, if you remember in the glycolysis video, you get 4 ATPs, right? And I believe you get 2 NADHs. I think it is. It's 4 ATPs and 2 NADHs. Right? That becomes 36 to 38 ATPs in this pathway. That's why I remember when we were dealing with cancer, it said that cancer is not as efficient. They said it's efficient because it's fast, but it's not as efficient as far as production is concerned because it only creates 2 ATPs per. Or 4 ATPs per glucose molecule, whereas the Krebs cycle or the full or normal cells, they were saying normal cells at that time, normal cells create 36 to 38 ATPs going through glycolysis or the sugar cycle. They just said glycolysis, I believe, right? Because it does not come into the Krebs cycle. That's what cancer does not do. And so because cancer does not do that, it doesn't deal with what? These issues that we talked about here, where it says, this is important. Remember, it says Cellular respiration is a multi-step process starting with glycolysis, which breaks down the 6-carbon compound of glucose and serves up the 3-carbon compound called pyruvic acids, right? And then it says what? It says first 2 carbons enter the cycle and 2 carbons are oxidized, removed from the cycle, right? That's the first thing it does. It gets rid of these 2 carbons, these extra carbons, right? We can think of this first step as completing the oxidation of the glucose sugar, and if we count 6 sugars enter the respiration pathway at glycolysis, and a total 6 must exit. But if they don't, through the Krebs cycle, they get stuck. And cancer is fast production, because you got to remember, regular cells is going through glycolysis and then going through this long cycle of 8 steps after that of Krebs cycle. So they're going through glycolysis to the Krebs cycle. Cancer ain't doing that. That's why it's growing so fast, because it's just going through that glycolysis and creating new cells. That's what it's doing. Stealing all your food, stealing all the sugar. So think about all the energy that it takes, that ATP, all the energy that it takes to make that a reality, and the oxygen that it's taking, because it's using aerobic glycolysis, right? Think of all the oxygen that is stealing, doing that, and not giving it back, plus leaving carbon in the body, undealt with, every cycle. You understand what I'm saying? So now you got to back up. You got problems all over the place. I told you that's a bad boy. Cancer is cold with it. I got to give it credit. It's cold with it. It is cold with it. It is cold with it, right? So now I want to come back. I'm going to come back to the chart, and we're going to use the other one, because I need to have this chart in front of you as we go through these steps, okay? So glycolysis. So okay, here we go. So cellular respiration is a 4-stage process. In the process, glucose is. Did I finish where I was at? I think I didn't. No, I didn't. Right. Where's the oxidized? 2 molecules are produced from each glucose molecule, so it turns in the Krebs cycle. No, we didn't do that yet. So hold on. No, I skipped that. We didn't need to do that. This we did need. So 36, yeah, there we go. 36 to 38 ATPs are formed from each glucose molecule, okay? But I do want you to see this, though. So Krebs cycle, no, it is a series of 8-step processes where the acetyl-CoA is oxidized to form 2 molecules of CO2, 2 molecules of carbon, and 1 ATP is produced. But it's going to be 2 of those right there, due to the fact that it's going to be 2 of those because it got to go through it twice, remember? Reduced high energy compounds NADH and FADH2 are also produced. 2 molecules of acetyl-CoA are produced from each glucose molecule. So 2 turns of the Krebs cycle are required. So remember, in glycolysis, 2 of these are produced. And when that happens, it has to go through it twice, which yields 4 CO2, 6 NADH, and 2 FADH2, and 2 ATPs. Now, NADH equals 2 to 3 ATPs. So you can do 6 times 3, that's 18. Right? That's 18 ATPs. And then you get 2 FADHs on top of it, and they are 2. So you could do that times 2. That's 4 more ATPs. And then 2 ATPs on top of that. Right? So this is how it's working. Now, so you're getting anywhere from 36 to 38 ATPs, which is extremely powerful. That's powerful, right? Because you're multiplying it by 2. So now, the 4 stages are glycolysis, partial oxidation of a glucose molecule to form 2 molecules of pyruvate, We know that. We watched that. I did a whole class on that. If you missed it, go back and see my mitochondria part 2. This process takes place in the cytosol, right? Not in the mitochondria, but in the cytosol. Okay. Now, next is formation of the acetyl-CoA or acetyl-CoA. Pyruvate formed in glycolysis enters the mitochondrial matrix, right? It undergoes oxidative decarboxylation to form 2 molecules of acetyl-CoA or acetyl. The reaction is catalyzed by the pyruvate dehydrogenase enzyme. Okay, this is the second step. So 2 pyruvates plus 2 NADs plus 2 COAs is going to produce 2 acetyl or acetyl COAs plus 2 NADHs plus a carbon. Okay, that's what's going to come out of this in that stage, and that's the first stage. That's the stage I showed you. I don't have to do that no more. That's why I did it the other way. Here, that's the stage I showed you here, right? So that's when it's gonna go into this stage here. Okay. 3. The Krebs cycle, TCA cycle, or citric acid cycle is the common pathway for complete oxidation of carbohydrates, proteins, and lipids as they are metabolized to the acetyl coenzyme A or other intermediaries of the cycle. The acetyl-CoA produced enters the tricarboxylic acid cycle or the citric acid cycle. Okay, glucose is fully oxidized in this process. The acetyl CoA combines with 4 carbon compound oxaloacetates to form 6C citrate. In this process, 2 molecules of CO2 are released and oxaloacetate is released. Energy is stored in ATP and other high energy compounds like NADH and FADH, which is good, right? That's what we want. We want that power. You want that energy. This is when you be like, I'm tired. You tired because this process ain't happening. That's why you need the balance oil. That's why you need to get your cellular respiration going. I'm out of energy. This is the problem. Electron transport system and oxidative phosphorylation. ATP is generated when electrons are transferred from the energy-rich molecules like NADH and FADH2, produced in glycolysis. Okay. Citric acid cycle and fatty acid oxidation to molecular O2 or oxygen, which is molecular oxygen, O2. Remember, if it's just O, it's atomic oxygen. If it's O2, it's molecular oxygen. By a series of electron carriers, O2 is reduced to H2O, which is water, right? That it takes place in the inner membrane of mitochondria. Okay? That takes place in the inner membrane of mitochondria. Okay? Now, Krebs cycle steps. It is an 8-step process. Krebs cycle or TCA cycle takes place in the matrix of the mitochondria under aerobic, in other words, under spiritual conditions. That's what it takes place under. If there is no spirit, there is no Krebs cycle. You got it? So, the first step is the condensation of acetyl-CoA with 4, and it will, believe it or not, it will try to keep going with the NADH that it has formed already. But what's going to happen is it's going to give off so much. Remember, this is the easiest way to say it. If you go back and you watch the Zenzino video, right? Remember when I talked about how waste can't get out and food can't get in, right? Remember that? Food can't get into the cell and waste can't come out. That's when this is stopped up. Because what's happening is it's trying to create without oxygen. And because of that, it's using the NADH, but the waste can't come out. And so you have massive waste product in there. And so Krebs cycle will stop when there's too much NADH or ATP just sitting there. The first step is the condensation of acetyl-CoA with 4-carbon compound oxalate to form CC6C citrate coenzyme is released. The reaction is catalyzed by the citrate synthase. So we saw that where? Here. Boom. That's step 1. Okay. Now, step 2. Citrate is converted to its isomer, isocitrate. The enzyme aconitase catalyzes this reaction, right? In other words, makes it happen. And this is what we, just real simple. So citrate synthase, right, is going to make this happen. Or excuse me, is going to make this happen. And then aconitase, right, citrate mixing with aconitase and water is going to make this happen. It's going to become isocitrate, okay? That's step 2. Now let's go to step 3. Step 3. Isocitrate undergoes dehydrogenation and decarboxylation to form 5C alpha ketoglutarate. A molecular form of CO2 is released. Isocitrate dehydrogenase catalyzes the reaction, meaning it makes it happen. What is hydrogenase? Dehydrogenase is a what? Enzyme. It's going to make it happen. It is an NAD plus dependent enzyme. NAD plus is converted to NADH, okay, which is extremely powerful, right? So it's going to be converted. So we see that here. Isocitrate then does what? NADH creates NAD plus creates NADH. Isocitrate dehydrogenase is going to kick off that CO2 or carbon dioxide that you need to breathe out. This is why breathing is important. And you have the alpha ketoglutarate made, right? Now, that's where we're at, right? Now we're going to step 4, I believe, right? Yeah, step 4. Alpha ketoglutarate undergoes oxidative decarboxylation to form succinyl CoA, a 4C compound. The reaction is, and I think that's what, a 4-carbon compound. The reaction is catalyzed by the alpha ketoglutarate dehydrogenase enzyme complex. One molecule of carbon dioxide is released and NAD plus is converted to NADH. So we're going to see that. So we got the alpha ketoglutarate, right? Then we have what? The NAD plus, right? And the NAD flips to a NADH through the what? Catalyst of the alpha ketoglutarate dehydrogenase, which also moves it forward, kicks out another carbon dioxide, which you need to breathe out, and succinyl coenzyme A is created. Okay? And we know if it's a coenzyme, it's going to help the what? Enzyme. Okay? Now, next, succinyl CoA forms succinate. The enzyme succinyl CoA synthetase catalyzes the reaction. This is coupled with the substrate level phosphorylation of GDP to get GTP. Remember, we talked about that GDP and we talked about GTP, which is ATP in another form, right? And GDP is ADP, no difference, just like ADP. Okay? It's almost the exact same equivalent, like I said, of DNA and RNA. This is coupled with substrate level phosphorylation of GDP to get GTP rather. GTP transfers its phosphate to ADP, forming ATP, just like it did in what? Glycolysis. Okay? Same thing. Now, succinate is oxidized by the enzyme succinate dehydrogenase to fumarate, right? In the process, FAD is converted to FADH2. This is no different than NAD being converted into NAD, or NAD plus being converted into NADH. This is just weaker, and it is, as I showed. Well, I didn't show you, I told you, but you're going to see in a minute. I'm going to show you in a minute. All right, in the process, FAD is converted to FADH2. Now, So that's step 6. So let's go look at that. Well, let's look at step 5 first. We didn't even look at that first. Hold on. So step 5 is succinyl coenzyme A, acetyl-CoA synthesis, or synthetase rather, is going to push this. Coenzyme A is going to get kicked out, which is good. Want that. And GDP, right, is going to go around to GTP, and then it's going to hit ADP. This arrow should actually be pointing this way. ADP is going to create ATP, right? We just read that, so you know it is. Now, the next phase that I read, which was step 6, I believe, right? Yeah. Is succinate, now using succinic dehydrogenase, right? Succinate dehydrogenase is going to now create FAD, which is going to kick out FADH2, which is power for the body, just like NADH and NADH, that's power for the body, right? To fumarate, okay? So it becomes fumarate. Now, after it becomes fumarate, then what's going to happen is fumarate gets converted to malate by the addition of one H2O, right, molecule. That's a molecule. So one H2O molecule. The enzyme catalyzing this reaction is fumarase, okay? So let's see that. So fumarate with H2O, which is water. Fumarase is the catalyst or the enzyme or what makes it happen, right? And then turns that into malate. Now, malate is the last step. Malate is dehydrogenated to form oxaloacetate, which combines with another molecule of acetyl-CoA and starts the new cycle, because remember, it has to go through it twice. Hydrogens removed get transferred to NADH plus, forming NADH. Malate dehydrogenase catalyzes the reaction. Okay, so let's look at that one time so you can see it. Malate, right? NAD plus is formed. Malate dehydrogenase kicks it into it. NADH is kicked out, which we use, right? And then oxaloacetate is created so the cycle can happen again. Okay, so it's a cyclic cycle. Now, and we're going to skip through that because we already looked at that. Now, Krebs Cycle summary. If you have a question at any time, please put it in there. There's no dumb question with this. This is not easy. Okay, it's not easy, but I want you to get it. I want you to understand it, because as we continue going through classes, as I continue teaching you about the different diseases and so forth, I'm going to mention this again, and you're going to get clearer and clearer and clearer on what's going on in the body. Remember, my purpose is not for you to be my client forever. My purpose is to make it where you do not need me anymore. That is my purpose. So when you will be a master of your own health, you will have perfected health. I don't know if you're the only one or not, but hey, welcome, J-Mo. Hopefully, a lot of times other people be listening, but they won't put nothing in. So yeah, but that's good, man. It's good that you're here. It's pretty straightforward. Good. I'm glad to hear that. And check out. Make sure you watch the other ones. So, mitochondria part 1 and part 2, because it's going to lead you up to this and you're going to really get it. Yeah, no problem at all. So, Krebs cycle occurs in the mitochondrial matrix. Acetyl-CoA, which is produced from the end product of the glycolysis, therefore pyruvate, and it condenses with 4-carbon oxaloacetate, which is generated back in the Krebs cycle. Okay. So these are the reactants. Okay, now Krebs cycle products. This is what happens afterwards, what we get out of it. Each citric acid cycle forms the following products. 2 molecules of CO2 are released, right? That's carbon dioxide is released. Removal of CO2 or decarboxylation of citric acid takes place at 2 places: in the conversion of isotrate 6C to alpha ketoglutarate, which is a 5, and I think that's saying carbon, 6 carbon or 5 carbon, in the conversion of alpha ketoglutarate 5C to succinyl CoA 4C. Okay, this is the two places it happens. When the carbon, is kicked off. Right, carbon is kicked off to go to 5C. 5C to 4C is kicking off a carbon. That's exactly what it's saying. That's exactly right. Okay, now 1 ATP is produced in the conversion of succinyl CoA to succinate. Okay, so an ATP is produced. 3 NAD plus are reduced to NAD, and 1 FAD plus is converted to FAD2 in the following reactions. So every time it goes through the cycle, we're getting 1 ATP, 3 NAD, H, right, which is going to be there. It's going to be 3 NADH and 1 FAD. So let's look at that. Let's see that. So we see we're going to get an NADH here, 1, an NADH here, 2, right? We're going to get an ATP here, 1 ATP, and we're going to get 1 FADH here. And we get another NADH. Is that 1, 2? Yeah, that's 3 NADHs, right? So we get 3 NADHs, we get 1 ATP, and we get 1 FADH2. Okay? That's energy. That's a lot of energy. And I'm going to show you why in a minute. I told you, but I'm going to show you. Now, following reactions. Isocitrate to alpha ketoglutarate is NADH. Alpha ketoglutarate to succinyl CoA equals a NADH, or produces, is what it's saying. Succinate to fumarate produces FADH2, and malate to oxaloacetate is NADH, okay? Note that 2 molecules of acetyl-CoA are produced from oxidative decarboxylation to 2 pyruvates, so 2 cycles are required per glucose molecule. That means during glycolysis, there are 2 acetyl-CoAs that are made. That's what it's saying. All right, so to summarize, for complete oxidation of a glucose molecule, each glucose molecule, Krebs cycle yields 4 CO2, right? 6 NADH, 2 FADH, and 2 ATPs. Now, this is what I was telling you I was going to show you. Each molecule of NADH can form 2 to 3 ATPs, and each FADH2 gives 2 ATPs. So NADH is stronger than FADH. That's why I was saying that. But GTP, it looks like, is equal with ATP. So NADH can form 2 to 3 ATPs, and FADH 2 ATPs on oxidation in the electron transport chain. Okay, so this is what happens. All right, so let's look at that and see if that's the case. And remember, each one can do 2 to 3 NADHs, and the FADH can create 2 ATPs. So that's why they're real strong. That's why they're good. So if we notice, we got 1, 2, 3 NADHs, which means, yes, that's 6, because it has to go through it twice. 3 times 2 is 6, as you know. ATP, 2, we're going to get 2 of those, and we're going to get 2 FADHs, right? And if we add all of it up and we multiply, it's going to be between 36 to 38 ATPs is the key. And do we get 4 CO2s? Yes, because we're going to get a CO2 here, and we're going to get a CO2. We're going to get one here. Is it on the other side? No, and we get one here, right? Right, because this becomes, so what's happening is it goes from a 6, to a 5, to a 4. And that's what's happening. And so we get those CO2s. When this transforms, we got to kick off a CO2 to get to the alpha ketoglutarate. And it's got to kick off a CO2 to get to the succinyl coenzyme, because they are carbon atom down. They're down at, I guess that's an atom. Yeah, that's an atom. Yep. And it creates a molecule of CO2, which is carbon dioxide. Yep. Okay. So, That was really good. Now, let's look at this. What hinders the Krebs cycle? What hinders it? The Krebs cycle can be hindered by a lack of necessary substrates like acetyl-CoA, right? Acetyl-CoA, right? So it can be hindered by a lack of this. A high concentration of ATP, which indicates high energy levels and thus slows down the cycle, right? That means it's backed up. Limited availability of electron receptors like NADH plus and FAD. Accumulation of certain intermediates like citrate and inhibition of key enzymes with the cycle by molecules like cicinol-CoA and NADH, depending on the specific reaction steps involved. So key points about factors hindering the Krebs cycle. High ATP levels. When ATP is abundant, the cycle slows down as it acts as a negative feedback regulator on several enzymes in the pathway. So if cancer is running rampant, right, it's creating ATP, but it's creating less ATP, but it's running rampant. It's massive of cancer cells, right? That can do it. And cancer does not go into the ATP cycle. It skips it. Low NAD plus and FAD levels. These coenzymes are crucial for accepting electrons during the cycle, so their scarcity limits the reaction rate, right? Because this right here, if you don't have these, NAD and FAD, you're going to have a problem. Now, if cancer is running rampant and you're not going through the Krebs cycle, right? Your cells aren't going through the Krebs cycle like normal, you're not going to have high amounts of NAD and FAD. As a matter of fact, no FAD is going to be produced at all, I don't believe, because it has to happen in the Krebs cycle. Enzyme inhibition. Certain intermediaries like citric can inhibit the citrate synthase enzyme, preventing further entry into the cycle. Okay? Enzyme inhibition. Oxidative stress. High levels of reactive oxygen species can damage enzymes in the Krebs cycles, impairing its function, and mitochondrial dysfunction. Issues with the mitochondria where the Krebs cycle occurs can disrupt the cycle's normal operation. So those are the things that can stop it. The other thing that will stop it is if it's not getting oxygen. So if it's getting backed up, it's not going to do it. If it thinks it's too much ATP, it's not going to do it. Right? Oxidative stress. This is why they talk about antioxidants all the time. Right? Oxidative stress, that's your mitochondria. So the balance oil is the key to the game with that. The balance oil, it is the key. So with that, if there are no questions, I appreciate everybody's time. Again, my book is out. Get the physical copy. Again, if you want it and you're in the Chicagoland area, I will sign it for you. If not, you can order it in the link below on the video, and you definitely will have it, okay? So with that, I appreciate everybody's time. See you all at the top of your health journey. Prayerfully, you got a lot out of this. Peace.
For the rest and more videos like these, you can join us at patreon.com/health class. Again, that's patreon.com/health class. And join one of our paid tiers. Any of them will do. And you join one of those tiers, you can watch the rest of these videos, as well as all the other videos that I produced, including my former health classes, which is 4 to 8 hours of information that can help you on your health journey to the top. All right. So I look forward to sharing that adventure with you. I look forward to seeing you at the top of your health journey. Peace.